Good morning slash afternoon to you, Ms. Jennings. This is my DNA project. So DNA replication is the process in which DNA makes an exact replica of itself. It starts out with the one original double helix shape consisting of nitrogen base, deoxyribose, and a phosphate. The three groups and stages of DNA replication process is the initiation, that's the first one. Then the second one is elongation, and the third one is termination. In initiation, it starts out In initiation, we start out with the helicase, which separates the DNA from each other because before it was one big piece and now it splits into two independent ones. Um, then an enzyme known as topoisomerase comes in and the application fork. And the main purpose of the topoisomerase is to make sure that the new DNA does not condense too much and it uncoils the original strand that the DNA is pushing through. A break that it once created, yeah. The next part of initiation are known as the single-stranded bonding proteins. They both attach themselves on each of the new separated strands that have became from the helicase. The function of the SSBPs is to make sure that the new formed strands do not reattach to each other. The main purpose of the initiation stage is to make sure that the replication fork is strong enough to make it through the whole DNA. If it is not strong enough, it is unable to pass through. The next part of DNA is elongation. And I'll skip to that right now because you'll see me placing the SSBPs, but that's not. I already said that. Okay, so it's elongation is when the leading and lagging strand are created. I added the leading strand and lagging strand a little on later, but I'll start with the main points. The main point starts from. I lost my words. My mom came down here. I think okay. So I start by making our leading and lagging strands. The 5' and the lagging strand runs from 5' to 3', and then that creates an enzyme known as a primaries or primase. I keep saying primary for some reason, but it becomes present on the leading strand. Then the primase is what creates the RNA primer, and that moves along the leading strand, telling and signaling the DNA to where to attach to newly placed DNA nucleotides known as DNA polymers. Primase then moves down to the lagging strand and does the same thing, but this time it moves from 5' to 3'. Then what happens in the lagging strand are known as fragments that are being made. The Zaki fragments are being made. Once the RNA primer moves down and create across the fragments, each fragment uses a different primer. The fragments are all separate. However, an enzyme known as ligase comes in between the fragments and connects all of the pieces together, such as DNA polymers, ligase. I'm trying to skip two so you see it. Ligase, and then RNA prime would be down there too. And ligase is like a glue in which it holds all the strong pieces together. Final step of DNA replication is termination, which the replication fork is being taken away, and the two forms of DNA are independent and are exact replica to each other. The overall goal of DNA replication is to create a replica of itself. Each stage has its own main purpose, such as initiation is to make sure that the fork and the strands do not come back, and the fork so they can go through. Overall goal of elongation is to make sure the parental strands unwind and the daughter strands are synthesized and are fully created and exact alike. Final goal is the termination, which it separates the two strands and makes sure they're both independent from each other and also that they're exact copies of each other. Thank you, Ms. Jennings, and have a good day/morning.